All right, thanks. So I guess Brian had a great introduction for what I'm going to talk about. Well, I'm going to talk about the unsupervised version of what Brian talked about two talks before this one. So basically, I'm going to focus on methylation data. And in principle, all of this could be done also for RNA, but this is only on principle because, at least in our hands, it doesn't work for RNA. So there's probably more work to do in order to get it to be done on RNA, at least with this approach. this approach. And the other thing is, the other difference is that we're not looking at sequence data, we're looking at array data. So basically, we're thinking about EWAS. You get a matrix, a big matrix, which is the rows are just the individuals, and the columns are just the CPG sites, so positions in the genome. So we have typically about 450,000 positions. In the new arrays, we have more, but most studies are done on these 450,000 by the number of samples. Typically, the number of samples that we saw in data sets are going between the hundreds and sometimes, just in a few cases, it's in the thousands. So right now, the matrices are small, but the cost is not so high, so it's potentially going to grow. How much time do I have? 30 minutes. So I'm going to skip this slide, okay? And I'm going to skip some of the introduction so that you'll get to the meat, right? So basically, we started thinking about it when we did a few years ago already, an EWAS on the differences between male and female. So we just, so essentially we get this big matrix, and our phenotype now is whether a person is a male or a female. So the difference between men and women. And when we run the study, so just if you run just, let's say, linear regression, you get all over the genome, you get great p-values of about, you can see here, 10 to the minus 200 sometimes, right? So that basically means if you're a man, you have high methylation, and if you're a woman, you have low methylation. It's always the case, essentially. Okay, so these could be either errors or they could be real. In some cases, we believe they're real, but the thing is, when we did that, we weren't sure. We wanted to check. So one of the things that you do in order to check, you basically do a replication study. We performed a few replication studies on different data sets, and we saw that we have a pretty good replication in the sense that we started with 11,000 associations after you correct for multiple hypotheses, and you end up with about 1,200 associations that were replicated in all studies. So I don't know if that's good or bad. The good thing is that we do have 1,200 that replicated in all 4 of them. The bad news is that we have about 9,000 that didn't replicate. And the question is essentially why? If we did all the study was done correctly, then why is this the case? So we try to think about what could go wrong, and so there could be batch effect, there could be differences in populations, there could be differences in demographics and so on. One of the things that could be different between women and men is the cell type composition. Okay, so that's basically what Brian talked about 20 minutes ago. I'm just going to go over this example a bit slower because I think it's key to understanding what's going on. So think about the following simplistic case. Let's say that you have 2 samples. One is a case and the other one is a control, right? And then you look at a specific methylation site, and that methylation site is on in 4 out of 10 cells, let's say, in sample 1, and it's in 6 out of 10 in sample 2. So obviously there's a difference. So let's say that it happens to all the cases. So all the cases have 6 out of 10, and all the controls have 4 out of 10. So now you can say, well, there's a big difference between the cases and the controls. The question is if this is actually because of that position in the genome, or is this because of something else that is going on? Okay, and what happens is that if you have, let's say, 2 cell types, one of them is a blue cell type and the other one is a red cell type, and let's say that that position is methylated in all the blue cell types, and it's never methylated in all the red cell types. what happens then? Then basically, if you have a difference between the cell type composition, between the cases and the controls, then you're going to have a difference in all the positions that are correlated, that the methylation levels are correlated to the cell type composition. So specifically in that position here. And so that's, even though it's true, there is a difference between the cases and the controls, that difference is not interesting in the sense that it's not, it's definitely not causal. It doesn't cause anything that is related to the phenotype. Okay, so it's indirectly related to the phenotype through the cell type composition. Okay, so in principle, we could just measure the cell type composition and that, just take the cell counts, and that's it. Okay, so if you run an EWAS and you don't take this into account, then you get a QQ plot that looks like that. So this is basically data on rheumatoid arthritis, where they basically ran an EWAS. And then what you see here, the x-axis, so this is a typical QQ plot. Basically, what you see here, what you're supposed to see is that so the x-axis is the expected p-value and the y-axis is the observed p-value. And you see that you have a lot of p-values. I don't know if I can, do I have, does this work? No. Okay, so what you see here is that all of these points here, they correspond to basically positions in the genome where you have inflated p-values. So everything looks like it's related to rheumatoid arthritis. If you correct for the cell type composition, which I'm not going to show you, but if you did, then basically everything would look like it's on the line, so on the line of y equals x, almost everything except for a few positions. So that basically means that most of the results that you see here, most of the hits here, are due to the fact that you have a difference between the cell type composition between the cases and the controls in rheumatoid arthritis. So obviously it depends on the condition. In some phenotypes you might not have such a striking difference between the cell type composition of the two, of the cases and the controls. Okay, so why does this happen? So let's get a little bit into the math and see why does this happen. So essentially what we typically do is a linear regression that basically looks at the phenotype Y and some covariates X. So that's just a matrix of covariates that could be sex, age, ancestry, and so on. And then you have a methylation site MJ, and you're looking at a linear regression of Y as a function of X and M. So you're trying to fit this linear model by finding alpha and beta. Okay, so the coefficient. Now, if Y is correlated with the cell type composition, okay, and the cell type composition is correlated with M, then M is going to be correlated with Y. Okay, so that could be that's going to give us this false positive. And one way to solve this is just to basically add the cell counts into the equation here. So basically we can add them into X. The problem is that in many cases we don't really have them. Okay, and some of the reasons Brian mentioned before. So one possible answer to this could be a supervised approach, which is, at least for array data, the most widely used supervised approach has been published in 2012 by Hausmann et al. And they basically do something very similar to what Brian mentioned. So some kind of a linear regression that basically takes into account the fact that you have some reference data from purified cells. Now the problem with this approach, so this approach is very useful in some cases, but the approach is limited when you, let's say when you go to a different tissue, or someone in the audience asked about a different species, but even if you go to a different tissue, you typically don't have the purified cells of, I don't know, if you look at kidney cells, okay? So that could be a problem over there. The other thing is that the reference is going to be specific. Typically, it's a very small sample size. So for array data, people look at six samples. So the reference is based on 6 samples. These 6 samples, first of all, it's a tiny sample size. The second problem is that it's a very specific sample size. It's a very specific population with a very specific age and sex and so on. So the distribution of that and ancestry. And it has been shown, for instance, that if you look at newborns and you look at their methylation data, then this method basically doesn't work at all. So correlation is 0. Okay, so and that's probably because the distribution in the cell type of the cell type composition in newborns is very different than in adults. Okay, so there's been a few unsupervised methods that people suggested. Some of them are more complicated than others. And actually, there's still all the time new methods that are coming up. I'm going to tell you a little bit about one of our method and some of its extensions. And our method is actually related to what you see at the top here, to PCA, Principal Component Analysis. And we do some kind of a variation of PCA, which I think that even if you don't use, if you don't do anything related to methylation, then maybe it's a good time to kind of like listen to what's going on here, because I think this could be useful in other contexts, as I'll show you, as I'll explain later. So other applications. So what is PCA? What do we do in PCA? PCA, typically, we have data in n dimensions, and we just make the assumption that there's some kind of a low-dimensional space where the data is actually lying in, and the rest is basically noise. And the noise is assumed to be normally distributed, and it's symmetric. So basically, all the directions are the same. So that's, if you think about PCA as a probabilistic method, so basically you have some kind of, you set up some kind of a likelihood, then this is essentially what it solves. It just assumes that the data is some, in this case, hyperplane or plane because it's 3 dimensions, but think about just a subspace of k dimensions inside an n-dimensional space. So what we noted is that basically if you look at the methylation data, then this assumption is too simplistic, because actually when you look at different cell types, then most sites do not change between the different cell types. It's only a small fraction of the sites that change. Okay? So basically the classical PCA model doesn't fit here. And what we suggested is some kind of a sparse PCA algorithm. So many sparse PCA algorithms have been proposed in the past, and hopefully I'll be able to tell you a little bit about how ours is different than others, or at least why we think it's better. But one of the reasons why it's better is because it's going to be very simple. So it's a 2 lines algorithm, and I'm going to give you in 2 slides. So again, basically the assumption that we have typically in PCA is that we have some kind of a signal matrix, which is plus noise. So noise is the further matrix over there, and then the signal matrix is in the middle, and then the result is what we have on the left here. Okay, and that's typically the assumption that we have, and you can see here that this is all obviously simulated data, but this is essentially the matrix in the middle. The signal matrix is a low rank matrix. That's why you see these kind of lines. What we assume in our model is that you have this kind of model. So you have noise, and then you have a signal data, but the signal matrix, but the signal matrix is 0 in most of the columns, and it's non-zero in a subset of the columns. Okay, so relatively small. So if you have 450,000 positions, let's say less than 10,000. Okay, so a small percentage. It's still not 1 or 2, but it's a few hundreds or a few thousands. So this is the algorithm. The algorithm is very simple, I think. Basically, what you do is you compute the low rank approximation for the original matrix M, and the way to do this is obviously using SVD, single value decomposition. So this is standard. Everyone that knows how to run PCA can run this. Basically, you just find the low rank approximation for the methylation matrix M. So this is what we call M tilde. And then we look at every column of M tilde and every column of M and look at the correlation between them. And we take so we give the score for that position based on the correlation between that position in M and that position in M tilde, right? So there's like two vectors now, and we can take the correlation between them, and then we take the T sites that have the best correlations. Intuitively, what it means is that we're looking at positions, at columns of the matrix M that can be explained well with a low rank approximation matrix M tilde, which makes a lot of sense to me. I don't know, maybe because I looked at it quite a lot, but essentially if the correlation between the low rank approximation of the singular value decomposition and the matrix M, the original matrix M, is very low, that basically means that we weren't able to reconstruct the matrix M at that column. Okay, so we shouldn't actually use it. Then we just run PCA on the top T sites. So that's the entire algorithm. So it's super simple. It's really just running singular value decomposition twice, and then running correlation between them. So 3 lines in MATLAB, 4 lines in R, 5 lines in Python. Something like that. So really, really, really short. Okay, so if you know how to run PCA, then you know how to run this algorithm. And in terms of runtime, it's basically dominated by the first step, which is running SVD. Okay, so again, that's just some kind of trying to give some intuition of why it works. Again, we have this noise plus matrix with low rank, which is the signal, and then we have our data matrix. And if we look at the correlation between column J in M and column J in M tilde, then this is kind of what we see, the graph that you see below, which is that's just simulation, but basically you can see that typically that correlation goes up. Okay, so on real data, we tried it on data where we had both cell counts and methylation data. So that data it was collected in order to study asthma on the Gala data from UCSF. And what you can see here is basically what the x-axis in each of these plots is the number of components that we use, and the y-axis corresponds to basically the R square between the true cell count and basically the estimation that we get. So if you go more to the right, then you have more degrees of freedom. So obviously it should go up. Okay, so you should get better and better results. But the absolute number is not so important. But what's important is the difference between the two lines, right? The PCA line and the refactor line. Refactor is our method, right? So you can see that typically refactor wins. In some cases, refactor wins by a lot. Some cases, they're, let's say in lymphocytes, they're about the same. But in all the other 4, there is a consistent win of refactor over PCA. Okay. So this is one way of checking this. We ran some simulations and we saw that that we get better results in PCA and a few other methods in the simulation. I'm not going to get into the details of these, but I wanted to mention one other way that we check this with some general conclusions. So basically, what we did is we took a few data sets where we had the methylation data, and we had either the cell counts, or we had estimates of the cell counts using the reference-based method. Okay, so and then we just believe these cell counts. Okay, so we have 2 matrices. The first matrix has just the methylation matrix, which is on the left. The right matrix is the cell type composition matrix, which is on the right. So basically, that would be a much, much narrower matrix, which has the number of individuals as the number of rows, but the columns are basically just the number of cell types, which is typically going to be a very small number, let's say 6. So you're going to have some numbers like this, and then the numbers on the right side on the cell types, let's say here there's only 3 cell types, and they sum up to 1. Now, the numbers are kind of the same in the right matrix, just to kind of make it simplistic, just for the presentation. But basically, they could be different. But the idea is that what we do is we cluster them. We cluster the individuals based on the matrix on the right. Okay, and we see that we have 2 clusters in this case. So we'll cluster them into 2. There's some kind of randomness in the clustering, which I'm not going to get into right now. Now we have 2 clusters, and then we define a new phenotype, which is going to be 0 or 1, depending on which cluster you're in. Okay? So note that, and that's basically our data, and now we're trying to find associations of specific sites with the phenotype. Okay? Now the phenotype was defined using cell type composition. So now if you use a method, let's say if you had a cell counts, okay? If you had a cell counts, you're not supposed to see anything that is associated with the phenotype, because the phenotype is defined by the cell counts, okay? That's a key point, so I just want to make sure that let me repeat this. So basically the process is that we define the phenotype based on the cell type composition, okay? So now if we add to the regression the cell type composition, we add the cell counts, then we cannot have a correlation between the phenotype and any methylation site, because that would be explained by the cell counts. Okay, so the p-value shouldn't be significant. Now different methods basically give different estimates now for the cell count. So if you look at our method, it gives some kind of PCs, right, instead of the cell counts. And if you look at let's say the reference-based method, so it gives an estimate which is not necessarily an accurate estimate of the cell count. And so this is what we get. I don't know if you can see here. The bold ones are basically the ones that win. So we have 6 data sets. And then we have, in each column, we have a different approach. One is just not to adjust anything. So basically not to use any method. So don't put the cell counts at all. And you see that in this case, you have quite a lot of, so every finding that we find is a false positive. So that basically measures how many false positives do we have. So you can see that in some cases we have thousands of false positives, right? So that's the leftmost column, right? Let's see if I can use this. Okay. So this column here is basically, you see that it's something. Now, in parentheses, that's what you get when you use FDR, and without parentheses, this is what when you use Bonferroni. So that's actually one of the things that you should take home, like a take-home message for this is not to use FDR, but I'll explain in a second why. You can use the reference-based with 7 cell types. And then in this case, you see that you have much less errors, but like you still have, in some data sets, you have even thousands of errors when you use FDR, and sometimes hundreds, and maybe the max is 1,000 errors when you use Bonferroni. Refactor, our method, still gives errors, and with FDR, it still gives sometimes a lot of errors. So in this case, it gives 2,200 errors. Okay, with Bonferroni, it's bounded by a few dozens. Okay, so basically, what you can take from this is that refactor is not perfect, but it's much better than what we see in other methods, and including actually the reference-based method. Okay, so this is very not always intuitive, right? Like when I started thinking about this, I thought reference-based method is basically the best you can get, right? But the reference-based method also has some problems, right? So first of all, it doesn't accurately find each of the, like I said before, it doesn't it's based on a reference data which is very, very small, 6 samples. But also maybe the difference between the cases and the controls is in a cell type that you haven't measured, right? Or that you have that somehow you have low counts from it. Okay, so then you're not going to get it in the reference-based method. Essentially, in all of these cases, we see that we have, except for one maybe, we see that we have an improvement when we look at the unsupervised method. And again, the reason why you get such an inflation with FDR is because FDR assumes that you don't have any confounders. And that's a very important thing to kind of keep in mind when you use FDR. When you use FDR, the assumption is that you took care of the confounders. In such a case like the methylation data and RNA data, it's also pretty problematic. You have a lot of confounders. And if you're not sure that the confounders are taken into account, then you're going to get a lot of false positives. And it's even worse because when you're going to replicate it, you're going to have sometimes the same biological confounders. Maybe not the same technical, but the same biological confounders. Okay, like cell type composition is still going to be a difference between the cases and the controls. Okay, one additional reason why we think, except for the fact that it's very simple and it worked well on methylation data, we have a rigorous proof that shows that under very reasonable assumptions, if this is your model, so noise plus signal equals your data, and the signal is column sparse, then essentially with high probability, which basically means always, almost always, so it's 1 minus something which goes to 0 when n goes to infinity, then refactor is going to give you better results compared to SVD. That's what we can prove, and our proof is limited in the sense that we make the assumption that the number of columns, of active columns that are on the left side are about n over log n, where n is the total number of columns. Okay, so that sounds like a small number relatively, but it's a fraction of 1 over log n. But first of all, if you think about what's going on in methylation, 450,000, if you only have a few thousand that are active, that's about the ratio that you see here. But also, if you try it in simulations, you see that it actually always wins. It's just that we only know how to prove it for n over log n. So there's a gap between the proof and practice. It always wins. What you see here, the y-axis is basically what's the error between the signal matrix and the estimated matrix. And the x-axis is basically how many columns are active. So I cautiously argue that it's always better to use refactor compared to just using PCA. In the worst case, it's just going to be the same. Okay, if it's not sparse. How am I doing with time? 5 minutes? 2 minutes? Okay, so I'm going to wrap up. So basically, one thing that I wanted to mention, and I'm not going to have time, but maybe I'm just going to briefly go over it, is that in some cases we want to do something which is kind of semi-supervised. So essentially, basically, let's say that you took cell counts from a small set of samples. So let's say 5% of your samples have cell counts, and the rest you didn't take cell counts. You can take this into account in the following way. So in 2 minutes, I'm not going to be able to get into the math, but basically you can assume that you have a prior distribution on the cell count distribution. And for that, to learn this prior, you don't actually really need the cell counts. You just need a separate data set that only has cell counts, doesn't necessarily have methylation. Okay, and these ones you could get with much larger numbers, right? And then you can learn the parameters of the priors, and basically then you can write down a likelihood model that would try to impute the cell types. Now, if 5% of your data is actually with cell counts, you can use these 5% in order to learn these parameters of the priors. And that's actually what we do. I'm going to show you this figure, and then I'm going to go almost to the end. So basically, in this figure, what you see is essentially the same data as before, the Gala data from UCSF. And here we assume that we know that basically we take 5% of the individuals that we know the cell counts for them, and the rest we just kind of ignore the cell counts, and we try to impute the cell counts. And you see that in some cases, granulocytes for instance, you get an R-square which is almost 1. Okay, so you get a very good imputation, even if you have only 5% of your study for which you took the cell counts. Okay, in some cases it's more noisy, and probably one of the reasons could be that you have less data. Okay, so I'm going to skip this because we're running out of time, but I just want to mention one thing that we did implement refactor and a few of the other methods that we did in kind of a command line tool that we call glint. And basically the idea behind glint is something very, I'm sure many people here are familiar with Plink. So it's very similar in spirit to Plink. If you can see here just highlighted here the type of command line. So you have basically command line and a log file that basically does everything for you in terms of the analysis. Once the data, so the one thing you do have to do is normalize the data. Once the data is normalized, you basically put it here, and then it does all kinds of things like the cell type composition, ancestry, and all kinds of statistics. It does some plotting for you, and I encourage you to go and try it, and if you have any feedback, then we'd be happy to hear. This was based on a bunch of papers that I got to, maybe 4 of them, but each of them like 3 slides. But and one important thing is that many of these papers were kind of driven by Elior Rahmani, that is a student of mine in Tel Aviv that is transferring here, which is, I think, we're lucky to have him. All right, thank you.